Ginger glue. Are you like, you keep pushing them on? When you push them
This one's
All right. Yale, you need
This one's
one of these.
I don't like that.
All right, y'all. Let's get started.
I'm cool like that. I'm a vampire and a leprechaun. Oh,
Let's pray. Father, the things that we've learned, would you help us to bring them to the front of our mind and diligence to study and concern about these things of life that you've created and the mystery of it and the wonder of it, Father. We are truly fearfully and wonderfully made. May we come out of this class with an awe of you, with a greater understanding of your work. Help all of our students just really pay great attention and make good sense of what's being presented. Help me to just be clear and compassionate in the teaching. We pray this in Jesus' name. All right, so what are our base pairing rules? You've been given a little thing to write up, right? Do you remember it? A something something and something something something. And I told you, hey, there's just so much information. If you just, now here's the thing, stop right now. That's good, it's better than it not being in our notes, but that's not the same as it being here. And what I really need is I need to go from here to here. Like, this has to be something we just know. Like, it just, boom, like, I will still sometimes have, like, students I taught 15 or 20 years ago go, I woke up in the middle of the night and I was remembering the DNA dance, or I was remembering like the hand signals for meiosis and mitosis, or I was remembering A2T and C3G, and I was thinking about nutty stuff because we just did it so much. We don't have the time to just do it over and over and over again, but it's got to get in there some way, somehow. It's got to get to the front of the mind, because on a test, that's where it's got to be. So A what? What does A pair with?
C.
A, okay, and what's the number?
2.
2. And that leaves C pairing with 3G. A2T and C3G. And what do we do with it from here?
We draw the circles.
Do circles. Right? Okay, good. And so that told us there's a lot of information here, right? So what are the base pairing rules? It tells me what A pairs with
C.
A to T, and C pairs with
D.
G. And G pairs with
C.
C, and T pairs with
D. A.
E.
Sorry.
A. Okay, good. Next question. Which bases are double ringed and which ones are single ringed?
A and G.
A and G are doubles, right? And T and C are singles. Okay, good. And which ones make single bonds and which ones make double bonds? It's up there. It's coded in that information.
Oh, A and T.
A and T make double bonds, right? So. Good. What kind of bonds are these?
Hydrogen bond.
Hydrogen bonds, right? Weak intermolecular forces of attraction, not covalent bonds. These are broken very easily, individually. But collectively, they're very strong. It's like Velcro, right? Okay. Next thing, describe the structure of DNA. What are some ways we talk about the structure of DNA?
Double.
Double helix. Good. What else?
Like it works in a pattern.
You tell me.
You have
Goes opposite direction.
Antiparallel. Good. I like that. What else? Think about the structure. Think about how it looks. How would you describe that structure?
like a ladder.
Like a ladder, okay? On that ladder, what's what? Think about a ladder. You got the two sides and you got the steps. What's what on this ladder of DNA? You got rails and rungs on a ladder. Rungs with steps, right? What are my rungs or my steps on DNA?
Those bonds.
Okay, they're in there, but what would you be stepping on if you're walking up DNA?
ATC.
Yeah, the bases, right? So, base, steps, and what are my rails, the side parts? What are they made out of?
The ones that connect.
Phosphates and sugar. Many times you've heard this from me: phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen. It's very rhythmical, right? When you do it like that. Phosphate, sugar. That's not for nothing. It's trying to drill it into that brain without, like learning without even learning. Phosphate, sugar, nitrogen base. This is the structure of DNA, right? We could just say this: phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen base. What are some other things we can use to describe DNA? What's it made out of? What's the sugar?
Glucose?
Not glucose. What's the D in DNA?
Oh, deoxy.
Deoxyribose. Okay. And good. What are my nitrogen bases? Phosphate, sugar, nitrogen base. Phosphate's phosphate, the sugar is deoxyribose. The nitrogen bases, there are 4 of them. What are they?
Adenine.
Adenine. What else?
Thymine.
Thymine,
Guanine and cytosine.
guanine, and cytosine. Okay, good. Good. Now the next thing on here says draw DNA with this particular strand. So I need to. You can do it. I think I gave you space, kind of to do it sideways, right? So what are these going to be made of?
Phosphate.
Phosphate and
Sure.
sugar. So I can go, all right, here's a phosphate. Here's a sugar. A phosphate. And a sugar. And a phosphate. And a sugar. I need 4 of these. Okay. So if you had something like this, where do my nitrogen bases attach? On the sugar. Phosphate, sugar, nitrogen base. And look. I even had, I tried to get you. I don't think you guys wanted to. You probably just thought I was stupid. But if you do it like this, hands up, bear with me, ready? Phosphate, point up, phosphate, then down, sugar, then in, nitrogen base. Phosphate, sugar, nitrogen base, phosphate, sugar. Like all of a sudden you're learning without even learning. Phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base. You kinesthetically, with your body, you're just learning, like, oh, phosphates only attach to sugars, but sugars attach to phosphates and to nitrogen bases. Phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base, phosphate, sugar. And then, like I said, I had them spin and get down low to the ground. This kills with 15-year-olds. I'm telling you, they love it. But you're too cool. It's all right. But you got to know phosphate, sugar, nitrogen base. So These attach. Right? Now, I gave you, what's our sequence here that I asked you for?
ATCG.
ATCG. Okay. So the first one I gotta draw is an A. What do I need to draw here? You don't say an A. I know an A. But what's it gonna look like?
2.
Yeah, double, right? Good. Good. And then this next one is T. Is that right? So what's that gonna look like?
Single.
Pardon?
It's just one circle.
One circle. Good. Good. What's this one gonna look like?
No.
This is C, right? So what's it gonna look like? 1 or 2? 1. What's this one gonna look like?
Two circles.
2 circles. It's G, right? Okay, good. Now, it's got a base pair. A pairs with
T.
T. How many bonds does it make?
1, not
2. T pairs with
3.
Hey.
A. How many bonds?
2.
2. C pairs with,
G.
say G. How many bonds?
3.
3. G pairs with C and it makes?
3.
Okay, good. So what do I put here?
T.
T, what's it look like?
One circle.
1 circle. And here I need to put
A.
A, and it needs 2 circles. And here I need to put,
G.
nah, G.
Two
And it's
circles.
got to have 2 circles. Good. And here I got to put
C.
C, and it's got to have 1 circle. Good. Now, I got to come in, I got to put a phosphate sugar backbone, right? Because it goes like this. Phosphate sugar, nitrogen base, phosphate sugar, nitrogen base, and you just get sick of doing this, but you get it. So, if this one is going this way, then this one must go
The other
this way, because they're anti-parallel. Okay? Okay. So I got to draw some sugars. Not too worried about the directionality of these pentagons right now. Do you know why I'm drawing them with 5 sides?
Because they have 5 bonds.
5 carbon sugars. All right, now, this is an A. What is this?
T.
A T. I can't draw a T like this T, can I? Because they are anti-parallel, so this T has to go this way. This is pairs with an A, but I can't draw that A. I got to draw this A because it's anti-parallel. Same thing here. Ooh, it's hard. Good. Okay. I guess I better draw these going the other way too. Something like that. That works. Either good.
So that's how we would draw, right?
Yeah, something like that. On a test, I'll have you go this way. It's a lot easier.
Okay.
And on a piece of paper, on a rake, like you guys don't have to go crazy like I am because you can just turn your paper. I can't turn this board. I'm not that strong.
Wait, I have a question.
Yeah, tell me.
On the top,
Huh?
wait, does it matter, like, which side?
What I really should have done, if I had it to do over, I'd have drawn these like this.
Does it matter which side the phosphate's hanging off of? Because on the top, it's on, but they're both on the same side, even though they're different.
You gotta be lost. What?
The extra phosphate, not the extra.
Oh,
phosphate,
well,
the
there's
one
gonna be
that-
a phosphate on both sides, if that's what you mean.
Well, yeah, but once it, does it just keep on going and going and going and never ends?
No, at some point it has what's called a telomere, which is the very end of the DNA.
And would they both end on the same, or would they end on different ones? On any side.
Like both with a phosphate or both with a nitrogen base? Is that what you mean?
No,
Or
I
any
mean,
sort of
with a, like a phosphate or a sugar,
I don't know the answer to
so
that.
it doesn't matter.
I've never had anyone ask that. And I've never thought to ask that myself. Okay, but the point is, for this, we're trying to just emphasize the anti-parallel part, right? That would be it. I'm taking this away. Okay. Next question. Draw the RNA from a DNA strand that read ATCG. So this is my, you know what my DNA looks like, ATCG. I want you to draw the RNA strand that would match this. What's it going to look like?
7T's over
Hmm?
here. 7T's here.
Okay, so this is the strand I want to pair with, right? ATCG, right? When you think about what happens with DNA, I have a strand, right? And then there's an enzyme that comes along, which I told you got to remember this, and you have notes on this. An enzyme called DNA helicase comes along, and it unzips this joke. Inside, I'll find A, T, C, G. Okay, this is my DNA. The RNA is going to be made by pairing right on top of this DNA, right? So right on top of this, A is going to pair with U, T is going to pair with A, C is going to pair with G, and G is going to pair with C. True or false, this is also going to be anti-parallel.
True.
True. So actually it won't look like this, right? What's it going to look like?
The letters will be upside down.
Letters are going to be upside down. So it'll look like this, and it'll look like this. It'll look like this, and it'll look like this. Do you have something that looks like this on the board presently? Yeah. It's going to look a lot like the complementary strand. There are going to be two major changes. One will be that A's will become, or I'm sorry, T's will become U's, and the other is that deoxyribose will become ribose. So if I want to draw, this is not what you want to draw, by the way. This is not it. This is just a step to show you. What it's actually going to look like is going to look like this, an awful lot like that, because of how RNA is produced. So, let's start out here. All right. Here, you guys should be able to fill this in, right? What do I put in each of these pentagons?
D.
No, ma'am.
Oh, R.
R for ribose, right? Good, so I'll go here. Okay. And then my T becomes
U.
U. And everything else stays the same, right? So A and G. And C. So my mRNA is going to look a whole lot like my complementary DNA because they're matching off of the same sequence of DNA. They've got to fit the same space, the same number of hydrogen bonds, the same shape, same everything. There are two differences between these things. Deoxyribose will be ribose, and T's will be U's. But otherwise, they are the exact same. All right. Next thing on here, what are the steps of DNA replication? So what's happening in DNA replication?
It has to be unraveled.
It has to be unwound. Do you know the name of the enzyme that does that? It's helicase. It's DNA helicase. DNA helicase unzips. Then the next step is, do you remember this? You cannot, the enzyme that's going to build
It
this.
has to hold it open, right?
Huh?
Something has to hold it open? Is that the helicase? that holds
Well,
it open?
so you have these molecules that hold it open. I told you you don't have to know those so much. Okay? But next, you need a molecule called RNA primase. RNA primase lays down RNA primers. There's just depending on your point of view, there's a deficiency in DNA in that it can't just start building itself out of nothing. It has to have an RNA primer to begin this. Then after the primer is laid down, then DNA polymerase lays down DNA. This all works really, really well on what's called the leading strand. So when I unzip some DNA, remember I want to copy both sides simultaneously. I'm going to make 2 copies of this thing. So as I'm moving forward this way, I can go. As it unzips, I can just go, 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 go. But this side, it's anti-parallel. It's got to work backwards. So as it began unzipping, I can go this way in this direction infinitely, right? But here I can only go as far as it's unzipped. So I have to repeat this process of laying down primer, doing DNA, laying down primer, doing DNA, and it's constantly going backwards, right? So one of these is going. Young, will you come up here? Come up. Needs milk. Okay. You be the leading strand, okay? So, come over here. Our DNA, we're going this direction. Now, note, remember, there's DNA going that direction too, right? But he and I are just going to go this direction. Come on, right over here. So he's going to be polymerase. He can get just You and I are too big for this. We're going to do this. Guys, we're going to bump into each other. You come this way. So he,
Oh, okay.
as the leading strand, he gets to work this way. And he just goes. But I have to work backwards because I'm going the other direction, right? So you go ahead and start. Come on, walk. And now I can start, and I can do that section, and as it keeps going, he just keeps going in it, but then I have to come, and I have to go this way, and I have to work backwards, and keep going. And then I have to run, and I have to, and I have to work backwards. I'm constantly, one side it just goes. It's easy, it's nice, it's smooth. The other guy's like having to work in these little chunks to catch up, right? So this goes smooth. This one is a chunk, a chunk, a chunk, a chunk, a chunk, a chunk, forever. Going in this direction, right? And it's the opposite over here. This one goes nice and smooth, but this one has to do chunks. Right? Fragments. What are these called? Okazaki fragments. Because everything sounds cooler in Japanese.
Why are we go Japanese? I thought we were in like I thought Latin was or Greek was the language we used.
This is not named after like a noun or verb in Japanese. It's named after Okazaki, the man who discovered this process. So, for the leading strand, this is all very, very easy. For the lagging strand with Okazaki fragments,
No, you stand there. You're gonna see better from right
I
there.
expect you to ace this Okazaki fragment section, right? For the Okazaki fragments, we have to remove the RNA primers, okay? And then we have to fuse the backbone together with DNA ligase. So, polymerase replaces RNA primers with DNA, and then ligase fuses the backbone, the phosphate sugar backbone. It creates covalent bonds between the adjacent phosphates and deoxyribose sugars. Right. Erase this side. All right. What are the steps of transcription? So what's the first thing I've got? This DNA, it's tightly wound. What do I have to do?
Unwind it.
Unwind it. So helicase unzips. And now?
Primase.
Yeah, they don't even need primase.
Oh.
RNA polymerase lays down RNA.
Wait, why don't you need primase?
I'm leaving in primase because. Check your notes. Did we have primase before? Maybe that I've forgotten something. That is entirely possible.
Yeah, I think so.
That's for RNA?
Wait. No, that's. Wait, did we talk about?
Oh, yeah.
I must have not written that one down. Oh, wait.
Well, let's watch it. We can do that. We have the technology.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot. This process occurs in 3 stages. In the cytoplasm of the cell, our cells absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into 2 processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons, code for...
We're not worried about introns and exons right now. We're worried about polymerase. So, many steps for replicating DNA. Super simple for RNA transcription, right? And that's what this is called, is transcription. So a transcript is a written copy of something that's been said. It's basically verbatim. Whereas translation, you're going from one language to another. RNA is the same essential language as DNA because they're both nucleic acids. But putting this into protein form, that's a whole different language, right? We're going to amino acids. So, let me change this back real quick. All right. Now, that's our next step here is describe the steps of translation. This is probably somewhere around where we left off last time. We talked about how there are 3 types of RNA. The one that we made here, this is called mRNA. It's messenger RNA. And what you need to know about messenger RNA is that it is a copy of a gene. Okay? It has what we call codons. codon is 3 base pairs. Okay. It's going to be read by a ribosome. All right. Then we have tRNA. This is transfer RNA. tRNA, first of all, here's its shape. It's very conveniently shaped like a T. Okay? It has what we call anticodons, and the anticodons will be down here. And the anticodons, the key thing with the anticodons is that they match it up against the codons of the RNA. Another thing about this is they will bring in an amino acid. These are essentially the link between mRNA and protein. Okay? So we take our DNA, make a copy of it into mRNA. We take that mRNA and I need to get it to protein. I need something that matches up with the RNA and that matches up with the protein. And that link is right here. Here's my RNA component that's pairing up and being read. And here's my amino acid. And these get used in a ribosome to form a protein. So it brings in the amino acids. It links. It is the link. I'm going to leave it at that. It is the link. If you don't have this, you cannot make a protein. You must have these. These are like the decoder rings of cell functions, okay? All right, and of course these are used in a ribosome. I'm out of room right here, so I'm going to just do this sideways. In a ribosome. Last type of RNA is very simple. It's just rRNA, and this is found. This is a structural component of ribosomes. You don't have to know anything more about this than that. Finishing that, I'm going to pull up a video to show us some translation.
So is that for translation?
Yes. Well, we're getting there, going into translation. So, to understand translation, one of the things you have to recognize, there are 3 types of RNA. There's the one that's a direct copy of the DNA that we're going to read. There's the tRNA that's going to help us to decode it, to go from the language of nucleic acid to the language of proteins. And then there's a third one that's a structural component of ribosomes that has to be in there to help it function. Okay? All right. Let's try this one. Oh yeah, it's okay. I need to do one more thing.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly-A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay. So what do you need to know all that? What must I have first if I'm ever going to make a protein? Gotta have some RNA. Gotta have some mRNA. Drawing it like this, it's not really like this. It's not spaced apart in threes. It's just a continuous strand of letters. Why am I drawing these in threes? The first one is going to be AUG, by the way, always. Because AUG is what's called the start codon. So these are codons. Codons are 3 base pairs of mRNA that code for 1 amino acid. I've got to have my mRNA. And the next thing is there are 2 units to the ribosome, right? There's a small unit, there's a big unit. Inside of the big unit, got to have tRNA, right? It's got to come in with an amino acid. And then the next tRNA's got to come in. The next amino acid. These have to get linked together. And as this thing strolls along, we're going to have more and more amino acid, or more and more tRNAs ready to come in with amino acids. Have to have a string of them coming, and this thing just moves along. 1 codon at a time. How many bases in a codon? 3. Right? The codon has to be paired with an anticodon. If my start codon is AUG, then what is the codon for the first, on the first tRNA?
U.
U. A.
C.
C. Good. So, in other words, let me draw it over here since I got a little more room over. AUG is going to have to pair with UAC. Those are literally going to be the bases on the bottom of this tRNA. Note the whole thing of this tRNA is RNA. Okay, and it's just bent and wound in a certain shape. And that one always codes for methionine, for what that's worth. You'll see that in a minute. Okay. So this is how it works. You gotta have your mRNA, you have a small subunit, you have tRNAs entering in, 2 of them will be in there at a time. You have your large subunit, and this thing just strolls along until it hits what's called stop codon. And then it releases. I don't need you to know the name release factor. I don't need you to know A site and P site. Okay? But you got to know about codons. You got to know about anticodons. You have to know the role of RNA. You have mRNA. You have to know the role of tRNA. You know that ribosome has 2 subunits that can attach. One interesting thing about this is if we're in dire need of a certain protein, you can have multiple ribosomes running along the same mRNA at the same time. And it can just mass produce stuff very, very rapidly. Okay? All right. How do you feel about this right now? Yes, no, maybe so? Okay. I'll have you good. I'm weird to ask. Let me just say it. Let me just encourage you. I post the videos and stuff like this up on the website. I post the lectures, so you can go back, you can look back through that, practice it, it'll help. All right. Okay, that was the steps in translation. Let's turn this thing over. Okay. Do me a favor, just write right on this, because it's set up that way, and it'll be nice and easy. All right. The DNA is ATT, TAA, CAG, GCA, CCC, GGG, ATT. Find the complementary DNA. So if the first thing is ATT, what's the complementary DNA? TAA. Write that in. And what's the next 3? ATT. Good. And what's the next one? GTC. Good. And give me a minute. And the next one?
CGT.
CGT, good. And the next one? GGG. The next one? CCC. And the last one? TAA. Right, these are just following our base pairing rules. A, what are our base pairing rules that we've learned in here? Letters and numbers? A, 2, T, and C, 3G. There's a lot of information coded in that. All right. Okay, so now I want you to find the mRNA codon. What is it? Which strand is it going to look like? The original DNA or the complementary DNA? It's going to look like the complementary DNA. There will be two differences, right? Structurally. One will be, instead of deoxyribose, we'll have ribose, and the others, instead of T's, we'll have U's. So, if my original DNA is ATT, my mRNA codon will be UAA. My second codon will be AUU.
Oh, yeah, sorry.
It's all right.
I was looking at the top one. I was looking at the second one, and I was like, it is perfect.
AUU. My next one is going to be GUC. The next one will be CGU. Then GGG, CCC, and the last one? UAA. Good. So just a reminder, the complementary DNA and mRNA are both coming from the original DNA. They're going to look, so those two are going to look the same because they have the same template. They're both opposites of the original DNA. But the tRNA is the opposite of the opposite. It's got to pair up with the opposite. So it's the opposite of the opposite. So it's going to look like the original DNA. Just food for thought, okay? So your mRNA codon, you told me, is UAA, right? So what's my tRNA anticodon? It's the opposite of it. AUU. And the next one, you told me the mRNA codon was AUU, so the tRNA is going to be UAA. If my mRNA codon was GUC, then my tRNA anticodon will be CAG. And if my mRNA codon is CGU, then my tRNA anticodon will be GCA. And if my mRNA is GGG, then my tRNA will be CCC. And if my mRNA is CCC, then my tRNA will be GGG. And if my mRNA is UAA, then my tRNA will be AUU. And again, I'll draw your attention to the fact that the complementary DNA and the mRNA look very similar. The only difference in the bases is the A's and the U's. And the tRNA looks an awful lot like the original DNA. The only difference is T's and U's, right? Just in case, let me draw this so that you can kind of see. I'll do it from what we have here.
When we're answering these types of questions, like, we won't, I mean, we can have in our mind that the sugar is R or D, but like, it'll be a lot of this, right?
Oh, yeah.
Just like, just the,
So this is my DNA strand, okay? So I might ask you to come up here and draw strand. So what we put here,
This is for RNA? Or the
we're
complementary?
literally doing this.
Oh, sorry.
So, don't be sorry if I said that, Mia, or in some way, I'm
No,
sorry. I apologize, I didn't mean that. I just mean like I'm actually just doing the one we have here. So if this is ATTTAA, then this needs to be
Oh, T,
uh-huh,
A,
uh-huh,
A,
uh-huh,
A,
A,
T,
T,
T.
T, right? Okay. Okay. And then my mRNA that I open this up so that I can make, right? It's got to be
U,
U,
A,
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 U, U, right? This is my mRNA. And then if I took this out and put it into the cytoplasm so that it was going to be red, and so here it is, and it's like U, A, U, okay? And now it's going to be read by ribosome. Then what? This is my first codon. What's my anticodon here?
A,
U, U, right? And then this has to be
U.
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 Ah, it's terrible. U, U, A, right? Okay. And, okay, so just a visual. This is my original sequence. The complementary sequence and the mRNA are going to be very similar. These are my differences. And then also, my tRNA is going to look an awful lot like my original DNA, except T's become U's. This should be UAA. I did something wrong somewhere. Get it here. That's my fault. Apologize for that. Okay. All right. And then we have one more question here, and this we haven't done yet. Okay. And it's how do I find the amino acid sequence? Okay. So I gave you a sheet, right? Take a look at that sheet. This one here. So this has 64 different mRNA codons. Okay, these are mRNA codons. So this is, hey, what does the mRNA sequence say on it? We want to know the mRNA, we want to know what amino acid is coded for by the mRNA sequence UAA. So what is that one? It's the stop codon. Okay. And what amino acid sequence is, or what amino acid is coded for by AUU?
Which, oh,
Isoleucine?
Isoleucine. Good. You guys see how you did that? Okay. What's coded for by GUC?
Valine.
Very good. And by CGU?
Arginine.
Arginine. Yeah, very good. Okay, and how about for GGG?
Glycine?
Glycine, very good. And for CCC? Maybe I'm having trouble
Yeah,
finding
that's
it.
right.
CCC?
Berlin.
Brilliant, very good. And then the last one is UAA.
What did you say? Listen. How do you say that?
Stop.
UAA should be stop.
Oh.
Okay.
Stop?
Yeah.
So that, is that a
that when you come across that, it won't put an amino acid there, it'll release.
Oh.
That's how it is to stop. Okay. Okay, so now the next several, I've given you different ones. The first one I gave you the DNA, the second one I gave you the complementary DNA, the third one I gave you the tRNA, and then the fourth one I give you the mRNA. Let's do just the first codon for each of these, okay? So if the complementary DNA on the next set is CGA, what is the DNA for that?
GCT.
GCT, good, write that in, everybody. GCT, right above it. And then if that complementary DNA is CGA, then what is the mRNA? You can either go to the DNA and find the opposite, right? Or you can remember that, hey, the mRNA and the complementary DNA are basically the same.
So it's going to be CGA.
CGA, yes, very good. Okay, what's the only difference between the complementary DNA and the mRNA? T is for use, good. So that's an E, I mean, literally, you could just write down that one if you wanted to. What's the tRNA anticodon? If the complementary DNA is CGA, then what is the tRNA?
GCU.
GCU, very good. And then go to your thing and find, for the mRNA codon CGA, what is the amino acid sequence? Arginine, very good.
We gotta memorize these, right?
No, never. Are you kidding me?
They
I have one of them memorized. AUG is start. That's the only one I know.
So are we gonna get this on the test?
Yeah. You crazy?
I had it
Oh my
in high
goodness.
school, but
There's like real stuff you need to learn. This is not it. Like you need to learn how to use this. You don't need to memorize this. You give me chills. All right. Let's do the next one. If down below. So if the tRNA anticodon is UUU, this one's easy, then what's the mRNA codon? AAA. If the mRNA codon is AAA, then what was the original DNA? TTT. And what was the complementary DNA? AAA. I like those. Those are easy. Let's do the one below it.
Wait, are we not going to find the amino acid sequence?
Oh yeah, find the amino acid sequence for AAA.
Lysine.
Lysine? Okay.
Oh, wait.
Yeah.
Why are there lines next to the finding amino acid sequence?
It should have been like a line all the way across just to separate these different like sets of problems.
Oh, well, there's, okay. So there's not, for the amino acid sequence, you don't have to put in the 3.
I'd write the 3 letters like GLY for glycine or
Oh, so we're gonna write
ARG
them,
for arginine
we're gonna write them like,
on those little lines.
all of, okay, yeah, that makes sense.
That's what I would do.
Oh,
Yeah.
never mind.
I'm giving you more here probably than I would on a test because this is practice and you need practice on a test. I just want to know if you can do it. I'll give you like 2 or 3. It won't be as bad. Okay, let's go down to the bottom one with the mRNA codon. If the mRNA is CCC, what was your original DNA? GGG. Good. And if your original DNA was GGG, what's your complementary DNA? CCC. And if your mRNA codon is CCC, then what's your tRNA anticodon? GGG. And if your mRNA codon is CCC, then what is the amino acid? CCC is proline, yes. Okay, so we got like 6 minutes left. Why don't you just work on this for these 6 minutes? Because let's be real, you're not going to work on it once you leave, right? So work on this, yeah, I know. So work on this for 6 minutes or whatever. I'll be here. Ask me questions if you have them.
So what else gonna be on the test?
The test coming up is only going to be, no.